Okay, so in the previous, the graduate student lecture, we went over current source density, derivation of the current source density. And now what we want to do is actually go over some actual data. So how do you actually record current source density? And more importantly, when you do these recordings, how do we come to the conclusion that we should represent the sources of current in the cortex as moving perpendicular to the cortex? So what is the actual data that supports the idea that when you look from a distance, you can summarize what's going on in the cortex as currents flowing sort of, perpendicular to either sort of, coming out of the cortex or going into the cortex. So we start off, where are we in the brain? So here's the brain. So here's an overly large central sulcus. So I have enough space to draw things. And so if we take just a little chunk of that, just a couple millimeters of that out, a cube out of that, and look at it, so look at it like this. So you got that little cube, and it's got layers in it. So here's like layer 6, layer 5, layer 4. So here's like 1, 2, 3, 4. That's the input layer. 5, 6. So, and then this has pyramidal neurons in it. So here's a pyramidal neuron here with some dendrites going up like that. So how can we actually sort of record from these guys? So simplest way to do this is just to put a sharp electrode into the brain. Now, when you think about putting an electrode in the brain, your first intuition might be, oh, it's going to cause a lot of damage when the electrode goes into the brain. But it turns out that when you put an electrode in the brain, you can think of what is the brain actually like physically? Well, it's this unbelievably dense, interwoven network of all these little processes going in every which direction. It's kind of like felt. It's like a million little hairs going in every which direction. And when you put an electrode into the brain, it actually does a very small amount of damage because it just kind of separates the little felt apart. Now you might think, like, what if you actually hit something? Like there's blood vessels in there. What if you hit a blood vessel? Well, it turns out if you take an electrode, like a sharp electrode, it's quite small in diameter, and if you aim it right for a blood vessel, like this, just move the electrode down, and when it hits the blood vessel, you think it would poke the blood vessel, but actually that's not what happens. Usually what happens is it just pushes the blood vessel off to the side, and it goes right past it, because the blood vessel is pretty strong. The same thing happens to many neurons. So unless you do an absolutely direct hit, if you just do a slight grazing hit, it just pushes it off to the side a little bit. And so commonly when you record, if you anesthetize an animal and do a bunch of recordings with a microelectrode, one of the things you might want to find out later on, if you kill the animal and section up the brain, to find out where your electrode was, you want to find out where was I recording from. It turns out if you just put an electrode in and pull it back out again, section up the brain, you can't find out where the electrode went. So it doesn't even leave any visible mark in the brain. And in order to figure out where where you actually recorded from, you actually have to make a little lesion by running some current through the electrode to make a small amount of localized damage that you can actually detect afterwards. So surprisingly, microelectrodes, they're very small, don't do very much damage to the brain at all. You can just sort of put it in and pull it out and almost nothing happens. So one easy way to record a current source density in 1D, so this is 1D CSD. So what do we need? We just need a whole bunch of measurements of the potential. And so here's our electrode. So we put our electrode here. So here's the electrode, and then it's going down into the cortex. So say we push it all the way down into the lower layers like that. There's our electrode. So, and then remember the electrode's all insulated. It's all insulated by a thin layer, so only the very tip is exposed. And so there's several ways of doing it. So one way, you could put an electrode in that has multiple contacts, but if you have multiple contacts, the problem is the electrode is going to be thicker, and it will do more damage. So one very easy way to do it is just put the electrode in, and then record at this point, and then pull the electrode up a tiny bit, record here, and just record it a whole bunch of different points until you pull the electrode all the way out. Now, what are we recording? So what we're recording is basically an evoked potential over time. So what does that look like? So last time I said, you're typically outside cells, so you see like a spike like there, and there's another spike. So the spikes are all upside down because you're outside of the cell. And then, maybe here's another spike on the way down. And so you see these large deviations with some spikes on them. And what is this? This is, voltage or, potential in that case, in that direction. And so what is in this direction? This is time. Now, typically what we do is we could either do a high-pass filter, which means save high, and then just get the spikes out. So now we just see like a little record of spikes, and usually we would flip it over so that they look like regular right-side-up spikes. So that's what you would do if you're doing single-unit recording. You just see the spikes. Or we could do a low-pass filter. So take the same signal and do a low-pass filter, in which case we would just get this big, this smooth change. And so this thing is what we're going to save. So this is called the local. We don't want the little spikes, which are just like, because we just happen to be really close to a neuron, and we don't want our signal to include the spikes. So this is called the local field potential. And that's mainly due to all these. So here's the spikes. So this is the guy that we're gonna save. So what does that look like? So now let's just. This is an actual 3D thing, but let's just consider 1D. So we'll just consider the depth. So now we'll simplify this down to just a one-dimensional picture. So here's a one-dimensional picture. And the dimensions here are time in this direction now, and depth in this direction. So here was depth before. But what was the other dimension? So the other dimensions up here, other dimensions were going parallel to the cortex. And there might be, for instance, a visual map when you go parallel to the cortex. So as you go in different directions across the surface of the cortex, you have representations of different parts of the visual field, for instance. We're ignoring that. So those are, so just for the time being, just ignore these guys. So now we're only paying attention to just depth and over time. So now we need to record our local field potentials in response to some event. So this is the event beginning. So that could be like a burst of a little tone burst, sound burst, or a little flash of some kind. It's just something that, it's some kind of stimulus that has a sharp beginning. And then what we do is we just do that stimulus over and over again every time we pull the electrode out. And even at a given point, we might do it, 10 times or 20 times in order to average. So here's an example. So there'll be some previous slightly noisy baseline before the stimulus started, after we averaged 20 cases of it. And then you might see, maybe something like this, some little deviation. So that's basically a bunch of samples over time at a particular depth. And then I just do the same thing at a different depth. So here's another sort of case here. So say this looks like that. Then here's another case, and say that one looks like that. And so there'll be a whole bunch of these recordings over time at all the different depths. So that's our raw data. So what is that data? That data is just voltages, and I've drawn. So say this is 0. So this is, voltage going in that direction, and then it could be negative also. So there's the 0. Say is the 0. So that's the graph for just one of those. But there's going to be another one over here. And another one over here, like that. Okay, so that's basically just voltage as a function of time, but also depth. So we've got sort of voltage going this way. We also have depth going this way at a bigger scale, because we've got a whole bunch of samples at different depths, and then time going this way. So that's our raw data. So that's our, voltage data. It's this guy. So now what do we do? So now we need to calculate the current source density. And so what is the current source density? Well, it's just the potential at a particular point in time subtracted, and then subtracting off the average of the potentials of the neighbors. That's how we calculate the second derivative. So what are we trying to do? We're trying to calculate at an instant in time the curvature. That's what the second derivative is, the curvature of the potential field at an instant. So let's just take this instant right here. So here's 3 values. So if we look at those guys, what do they look like? Well, if we're calculating the current source density for this guy, this one was a little bigger, and that one was a little bit bigger. And so when we subtract off the average of these guys from this guy, we get a negative number. So that thing indicates a sink. So that's the voltage, and now this is our 1D current source density. So as a function of time. So here's time again. And so this guy, after we do this calculation with this guy and that guy, we're going to just end up with a single number. Did I make that one? Yeah. That guy goes there. So a single number, and that number is going to be negative. So this guy, the result of this calculation goes into one number. So that's how we calculate the current source density, and you just keep doing it for everything else. So notice it doesn't depend on time. So how do we calculate the next one? Well, we just go, a slightly later point in time, get those numbers, and then we get another number for the current source density. So the current source density, so what will happen is you'll end up with some other little wave like that. Except now, so our calculation, all we did was just take the second derivative of the potential, and we get the current source density over time. But our calculation is just instantaneous. We just do it for each instant in time as if it doesn't matter what came before and after. And so then you'll get a whole number, you'll get another set of curves here. But these are kind of more valuable curves. And so why are these curves more valuable? Because now, at each point in this curve, if you're up like that, that means a source. And if you're down, that means a sink. So now we have something more valuable than just the potential. And if we look at this one right here, I drew it this way in particular to show what the extra information is. So if you look at this potential here, so like an evoked potential, imagine you've got an electrode on the scalp, you get a positive voltage relative to a reference, like your earlobes or the base of your skull or something like that. So you get a positive number. But that positive voltage doesn't really tell us whether there's current sort of erupting out of that point or disappearing into it. And in fact, because the electrode above and below are higher, it actually indicates that current is disappearing there. So even though it's positive, even though the potential is positive, the actual, try that in the same color. So this is the voltage, and this guy is I'll use that symbol. So this is the 1D current source density. So even though the potential at that point, the voltage at that electrode is positive, it actually corresponds to a sink, meaning that the current is disappearing there. And so it's relatively difficult to eyeball this just looking at these plots. The same way with evoked potential. So if you say, I've got an N1 or something, like I've got a negative number here, in some respects that's not very informative because it really depends on what's around the N1 to determine whether that's actually a source or a sink. So that's how you would do this in 1D. So it's a very straightforward thing. You just do a whole bunch of samples, and then you just do these calculations at each point, at each instant in time by comparing to your neighbors, and then you just get whether you've got a source or a sink. So what can we sort of conclude from doing these kinds of experiments? So the first thing, before we talk about like these other dimensions, let's just talk about if we do these experiments and sort of make a cartoon of what this data actually looks like. So I have in the reading some examples where people have done this. This one happens to come from I happens to come from a click in rat auditory cortex. So rats can hear pretty well. And so if you just do a click for your stimulus, that stimulates a lot of different parts of the auditory cortex, and then we can look at how the current flowed over time. And so the picture you get looks something like this. So I'll draw it over here. Okay, so here's the cortex depth, and now what we're going to do is just sort of color things in to indicate sources and sinks. So here's layer 4. It turns out it's relatively hard to see sources and sinks in layer 4, and the reason is because layer 4 often has stellate cells in it that are not like long pyramidal cells, and so they're a little bit more spherically symmetric, and so that those tend to cancel out some of the large signals. But if you do a whole bunch of current source density experiments in 1D, you get something that sort of looks like this. Okay, and so I'm coloring in sources and sinks. Okay, so which is sources and sinks? So this is, the open ones are a source, and the dark ones are a sink. So remember, this is time here and depth. So overall, what do you see? So overall, what you see is you first see a source going to a sink toward layer 4. You actually don't see the initial. Remember what we said is that layer 4 is the input layer, so stuff comes into layer 4, and then it synapses on cells in layer 4, and they sort of go outside layer 4, like that. It turns out you actually don't see this. You don't see much of this initial excitation. So here's the the input coming in. So this part, unfortunately, you can't see very easily. It's the most, in some respects, the most important part. What is the input to the direct input to the cortical area? And the reason is because the cells are not so elongated there. But soon afterwards, you see a source going to a sink. So you see effectively a current dipole flowing from like above layer 4 down toward layer 4. And then a little bit later, you see the opposite. You see kind of like you see a current dipole flowing from the lower cortical areas going upwards. And then you see another one that sort of goes down like that. And then sometimes you'll see a fourth one that goes up. So that turns out to be a pretty characteristic picture of how many different cortical areas respond to a brief stimulus. It has this characteristic sort of sort of source and sink going over time. And what do we think that actually corresponds to? So let's just draw the first 3. So here's our guess as to what. So this guy is that one, and this guy is that one, and this guy is that one. So what do we think is actually going on in the cortex? So we said that it's a little hard to see that first input coming into layer 4. So here's layer 4. That's the input layer. So it's a little hard to see that, but what you do see are some big pyramidal neurons that there are some pyramidal neurons in layer 4 also, in addition to all the little stellate ones. So we can't see the stellate ones very well, so those come in and kind of synapse on these guys, and then these guys go up and synapse on these guys. So can't see this very well, but we do see some current going in here and coming out down here. So that's this is basically our oh, sorry, I got it the wrong way. Sink going to source. Yeah, okay, right. I've got it the right way. Yeah, I always have trouble with this in my notes. I should. So this guy's a source up here. This guy's a sink. So our current source density basically shows a current dipole coming from the currents that are outside the cell. Because remember, like when it's inside the cell, it's kind of disappeared and we can't record it, and we're always recording outside the cell. So it's the reverse. So we don't see the current's actually coming in at the basal dendrites, flowing up the cell and coming out the top. So it's erupting out of the top and going in, it's disappearing here. And so when we draw this as the source is generating something and it's going to the sink. It's the return. So basically what we see return currents. So I confuse myself every year, but I didn't. I recovered this year, So basically we see a source going to a sink, which are the return currents, the currents that we can't see because they're invisible because they're going inside the cell and we're not recording inside the cell, are basal dendrite channels opening up, current going into the cell and flowing upward and then erupting out of the top of the cell. So that's the explanation for why we see a source here going down to a sink. It's the return currents that we're looking at. Okay, so what happens next? Well, then we've got some big cells down here, like say big motor cells that are going to the colliculus and are kind of like the output, somewhere in the output layer. Drew that a little bit above the output layer, but let's say that was a big layer 5 cell. So here's layer 6 and layer 5. So now what happens is we see these guys being stimulated in their basal dendrites, and the current going through the cell and coming out here. So basically now we're seeing got it the wrong way. Coming out the basal dendrites, so being stimulated at the top. And so this means that this guy is a sink because current is disappearing into the apical dendrites, and it's a source here. And so that means our current dipole is going that way. And that's this guy. So the return currents, the source of the return currents are down here, and they're going into the sink up there. And then a little bit later on, you have guys that are in both of these things being stimulated on their apical dendrites. So now they're stimulating on their apical dendrites like that. And so that boils down to a oh no, I drew them the right way in my notes. Yes, they're being stimulated on their how many? Okay, going that way. Okay, so we see a source up here, sources, and we got sinks down here. And so that corresponds to something that's going that way. And so what do we call these things, like in the evoked potential the evoked potential literature. This is the P1. So the positive 1. So notice that this is classifying it by the sign of the potential, which is not a good thing to do because it turns out that a P1 could be generated by an overall negative voltage. But this corresponds in time to what an evoked potential person would call the P1. And this one corresponds in time to what an evoked potential person would call an N1. And then this is the P2. So these are extended quite far in time. We're talking 100 milliseconds, spreading out over a couple hundred milliseconds. So this is digging down to the level of an individual cortical area, ignoring like the two-dimensional structure of it, and just looking at what's happening as you go down through the layers. This is the kind of pattern that you see in many cortical areas. And it reflects that cortical cartoon that I showed you. Input comes into layer 4. It then spreads particularly to layer 2, 3. So this guy, we'll put the synapse down here. It spreads particularly to layer 2, 3, and then later on there's some stimulation of these big apical dendrites that are the kind of the output structure. So you basically sort of stimulate the basal dendrites, and then you stimulate apical dendrites, and then you stimulate basal dendrites again, and you get this oscillating pattern. And that's present in every cortical area. And so you can see partly what the problem is. If all these cortical areas are generating this, this kind of structure like this, and here's another cortical area, higher cortical area, it's also generating that structure. And there's another cortical area, it's also generating that structure, and you add those all up. And then you get your scalp ERP. What does that reflect? That reflects basically all the cortical areas all doing the same thing approximately with slight offsets. So you can see why it's very not straightforward to just eyeball evoked potentials like many people in the evoked potential literature do. They just like pick out, they take a scalp electrode and pick out, that's the P1 peak. Yeah, we'll see that's not the deepest way of looking at what's actually going on here, because what's actually going on is all these areas are doing pretty much the same up and down and up and down. Okay, so that's 1D. So we have a little bit of time for 2 and 3D. So one of the things I said at the beginning was this kind of current source density analysis has really convinced us that we should model the cortex as these current dipoles. And why is that? So the critical observation is, if we do the same thing at a different point in the cortex, so let's put another electrode in over here. So go to a different part of the cortex and record a bunch of evoked potentials at a bunch of different. So here we've moved in this map. So if you do that, it turns out. So if we move this. If we basically look at 1D CSD moving in map. So we typically have, so if we move in the map and record a different 1D current source density, what do we see? So the result here is that very slow change. So slow meaning like if we move a millimeter, not much happens. So very slow change in 1D CSD. So when we go through when we go through the cortex, even if we move like say 50 microns or 100 microns, we move like a tenth of a millimeter, and we see a sudden change, like you might see something like this. I just moved a tenth of a millimeter, and suddenly the sign of the voltage changed, and the current source density calculation had a big change. I didn't make a big change there, but there's supposed to be a big change here. Let's make a big change. Okay, make a big change there. So when we move in the map, we get very slight, much more gentle differences in what the current source density looks like compared to the changes we get when we go through it in this vertical direction. And so that's a fundamental observation that shows. So that's how we mainly conclude. Therefore, local perpendicular current dipole is, a good approximation. So that's the basic observation. And this is true in all different cortical areas. So if you just go within V1 or auditory cortex or a higher level cortical area and do current source density and then move the electrode to some other part of the cortex and do it, you'll always find something that's actually very similar if you just go over like a millimeter compared to just moving down a 20th of a millimeter and you get a sudden change. So that's basically the rationale for why we should model these currents, model the sources and sinks of currents as just moving perpendicular to the cortex. Now, of course, we know these cells are, they're complicated cells. They've got, dendrites going all over the place. There's current going sideways through the dendrites. There's axons, that come and run this way, and synapse on things, moving side. There's a whole bunch of that going on. But the overall picture from the ground truth current source density is that we can sort of approximate that just as a current flowing vertically in the cortex. So it's important to sort of see the rationale for why we model things at the higher level as just a current, a perpendicular to the cortex current dipole. Okay, now what if we back off a little bit? So if we back off a little bit and go to a larger scale, well, then we can start to see differences between different parts of the cortex. And so one thing we can do is we can do a 2D current source density. So that would basically be record a whole bunch of samples, but just moving parallel to the surface of the cortex. And so you can do this in the cortex itself by just putting an array of electrodes on the cortex. So instead of doing this, what we can do is we can just put like a sheet of electrodes. Just get a tiny little flexible sheet of electrodes that has like a whole bunch of contacts on it. And then those all sort of go off to a big bundle of wires. And then they can have little nibs sticking down so they can touch the cortex. So we have all those we just lay that sheet of electrodes on the top of the cortex. And they'll be spread out further apart than this spacing here because we're looking for this larger scale picture. But if we do that, what we can see is that, so we'll just have a whole bunch of samples spreading out over, say, a centimeter. Something like that. And then what do? We, look at all the neighbors and then figure out, where you are compared to the neighbors. So compare that guy to all its neighbors, subtract off the average of the neighbors, and that will give us our current source density. But now it's a 2D current source density. So this is 2D CSD. And what are we sort of assuming now? Now we're assuming that all the cortical areas behave exactly the same way. It's not true, of course, but they all, their pattern of sources and sinks after a period of time is the same for all of them. And so now we're just looking at what are the differences between different cortical areas moving in this direction. So that would be, this would be sort of for time t1, and then we would do our Laplacian calculation, which is take the second derivative of that guy. And what does that give us? That gives us the current source density. So this guy will, after we figure out what it is relative to its neighbors, we'll get a number down there. And so what happens in this case is that the picture that you get here is going to be a little more complicated than this one, because, for instance, just as a simple example, say we had something like this. I'll draw a whole brain, and imagine we're doing it on the scalp. So here's the skull and the skin, and here's an electrode up there, another electrode up here. Now, let's say that our current source density, our local field potential at a point in time, look like something like this. So this is the potential, local field potential at point in time. So that's all positive. So if you just eyeballed the local field potential, you just see sort of a peak over here. But you can see at this point, if we compare this guy to his neighbors, he's less than the average of his neighbors. And so that means that at that point there's actually, at this point there's actually a sink. Current is sort of disappearing into the scalp there. Whereas this guy, compare that one to its neighbors, that guy's a source, and here's another source over here. So generally, when you look at the current source density, so this is the potential, and here's the Laplacian of the potential. When you look at the current source density, it's going to be more complicated. Now, that doesn't tell you what's actually causing this. So it could be that, it could be that there was a cell, something like this. Let's move it over here. So maybe this part of the thing came from the fact that some current sort of, disappeared over here and then came out over here. But, it could also be something else. It could be that, there were two cells and, current disappeared into this one and current came into this one and went up and then came out here. So you could have sort of multiple situations that could have caused this. And this is just like an approximation of what would have happened if the cortex was just a little thin rind right underneath the skull. So it's not a way of doing a good job of localizing where the currents are coming from, but it's better than just looking at the potentials, which is what most people do. So once again, this is a calculation that you just do at each point in time. So here's, time t2. You can do the same thing over here. So you got another sample, and then you look out at the neighbors at each point, and then you use that to, calculate another point in time down here. So what you're going to see when you do a current source density is, say you might have had sort of a positive field here and a negative field here. Say maybe that's the way it was, positive. But the positive could be something like this, where it's got a little divot in it. If it has a little divot in it, it's still positive, but that corresponds to a sink, not a source. And so then what will happen is you'll often end up with a much more complicated picture, sort of more informative picture in a way, because it's telling you which cortical areas are. So it might be positive here, current source density, negative here, positive here. And you could have the same thing, occurring down here, negative, positive. So you'll see a more complicated spatial pattern of current source density, more informative pattern of currents of where the currents are going than you would if you just look at the potentials themselves. So there's still a lot of ambiguities, because we know that if you excite the basal dendrites and positive current flows up, that looks the same as inhibiting the apical dendrites with a negative current flowing down. So you can't tell inhibition from excitation. You've got this problem of well, what if the neurons were sideways versus vertical? That's going to be a problem. But in general, I would say it's a good thing to do, because it's better than just looking at the bare potentials, because it's giving you some kind of estimate of where the currents are coming out and going in. Now, it's best if you do it on the cortex, but obviously with a human subject, you're probably not going to put a little grid of electrodes on the cortex unless you have like an epilepsy surgery. And so, but you can do the same thing on top of the scalp. And what's going to happen, of course, is that it's going to blur out, because some of these currents they're going to prefer not to go through the high resistance skull, and so they'll flow, they'll flow some distance before they come out. But nevertheless, more of them will come out right here, or go in right there. So it's still a good thing to do on the scalp, to sort of get a better spatial picture of how the currents are flowing. So that's and then last but not least, in 30 seconds, you can do the true gold standard, which is 3D current source density. You can see what the problem is here. You need a whole ton of electrodes. But it is possible to do it, and I've got a paper in the readings that did it. And so what you can do is you can just take a chunk of the brain and then just put electrodes down in all different parts of the brain and record a whole bunch of points in 3D. And that's the real gold standard. Then you would say, I know exactly where the currents went in this part of the brain. But you can also do the 1D going through the cortex to sort of figure out like that sources and sinks vary quite rapidly as you go through different layers, or 2D, which shows you that sources and sinks vary much less rapidly, but nevertheless do vary, and that's informative as well. But if you really wanted to localize with current source density, you would have to do 3D current source density. Okay, so I think we'll end there, David, and I'll take some questions.